Hello, I am Dr. Azaz from medicovisual.com, and in today's visual lecture we will discuss in detail the endochondral ossification of the bone. To understand this lecture, I will highly recommend that you first watch the previous two lectures of this lecture series. Basically, this is the third and last lecture of the ossification of bone lecture series. If you are watching on YouTube, I will put a playlist and I will give the link in the description and you can watch the playlist. In that playlist, you will see that first video is about the basic differences between intramembranous and endochondral ossification. Then in the second video, we had talked about the intramembranous ossification, and in this video I will discuss the steps of endochondral ossification of the bone. Now in this video I will give the reference from the previous lectures, especially from the lecture of intramembranous ossification of the bone. So that is why it is important that you first watch those lectures. So starting from the very basic concept that, as we have discussed previously, that in the intramembranous ossification, there is the formation of mesenchymal membranes, basically, which are condensation of mesenchyme, and these mesenchymal condensation directly from the mesenchymal condensation directly bone forms. But here there is an extra step in case of endochondral ossification. There is an extra step that, of course, here also there is mesenchymal condensation, but these condensates, they are first converted into cartilage, and then this cartilage is step by step, gradually, it is not converted, but it is replaced. It is replaced by the bone. And this is the final bone that forms via the process of endochondral ossification. Now the thing is, the interesting thing here is that the bone that develops here, actually whole of the bone that develops here, it actually develops with the process of intramembranous ossification, and the cartilage is basically acting as a mold, or it is acting as a template which guides the bone, which guides the formation of proper structure of the bone. I know these things might confuse you right now, but as we proceed further with this lecture, these things will make a perfect sense. Don't worry about it. We will have a proper visual learning experience in this lecture as usual with the medico-visual. You are used to it. So let's start the video. So let's start from the very basic concept that we have these mesenchymal cells, and these mesenchymal cells, although here they look like that they are leaf-like, but actually they are not leaf-like. These are irregularly shaped cells, mesenchymal cells, and these are scattered, these are interspersed in a gelatinous ground substance. And this is basically the fundamental and foundational material from which the bone develops. So now this is basically called mesenchyme, and mesenchyme is basically nothing more than the embryonic connective tissue. So this embryonic connective tissue, that is mesenchyme, it condenses, these cells, they come close together, they cluster together, and they form a dense shape that roughly resembles the future bone. So it roughly forms the shape of the future bone like this. So you see these mesenchymal cells, they came close together, they huddled around each other, they hugged each other, and they roughly form the shape of the bone. This condensate, it formed roughly the shape of the bone. Now these mesenchymal cells, some of them might die out, but those that remain, they will transform into chondrogenic cells, or they will transform first into chondrogenic or chondroprogenitor cells, and then ultimately those cells will form chondroblasts, and then chondroblasts will convert into chondrocytes to form the hyaline cartilage. I will not discuss these details because that is the topic of development of cartilage, and it is not very relevant from the exam point of view and from the clinical aspects as well. So we will just ignore it. But the thing is that you must understand that this mesenchymal condensate, it will transform, it will not be replaced, but it will be transformed into cartilage. Compare this with intramembranous ossification, where this such type of condensate they form and they are directly transformed into bone. Here they are not being transformed into bone, but they will be first transformed into cartilage, and then that cartilage will be replaced by the bone. So hyaline cartilage develops. So as I've told you that chondroblast, these cells, some of these mesenchymal cells, some of them die, the other that remain, they will transform, they will differentiate first into chondrogenic cells or chondroprogenitor cells, and then those chondroprogenitor cells, they will further differentiate into chondroblast, and then chondroblast, they will then form the ground substance of the hyaline cartilage, and then they themselves will form the chondrocytes. So in this way, this glassy material, this glassy beautiful cartilage forms, and this cartilage is called hyaline cartilage. Now this hyaline cartilage is actual template or mold of the bone. So, and of course mesenchymal cells, they will also be surrounding these the hyaline cartilage, and they form this perichondrium around it. They form the perichondrium around it. So here you can see this one is the perichondrium. Now here you see that perichondrium does not surround the hyaline cartilage in this case from all sides. Some of these sides, they do not have the perichondrium. Second thing that you must understand, that you must remember about the hyaline cartilage, is that this hyaline cartilage, it does not have blood vessels. Some blood vessels may be there, rather I should say some blood vessels are in fact there within the perichondrium, but blood vessels are not inside. Blood vessels are not inside the cartilage. Inside the cartilage here, there are no blood vessels. So what actually happens, that the nutrients and oxygen that diffuses into the cartilage. So these nutrients, they should diffuse into the cartilage. There is no blood vessel that will take the nutrients inside the cartilage. This is important point because it will make some sense later when we will discuss the cellular hypertrophy and cellular death and stuff like that, right? So this is the basic concept that cartilage forms. Now this cartilage, it will grow in size, it will increase in size. So this cartilage will increase in size, and it increases in size by two methods. Let me briefly tell you, because this is important and it is mentioned in the literature. So here, if I show it here, that basically here is the perichondrium, and perichondrium here consists of, the inner layer of perichondrium is cellular, the outer layer is fibrous, and inner layer is cellular. Now this inner cellular layer, it contains the chondroprogenitor cells, and chondroprogenitor cells, they continue to gradually transform into chondroblast and then chondrocytes. So this here more layers will be added from the perichondrium side, more layers will be added. So thickness of the cartilage will increase. This type of growth of the cartilage is called appositional growth. With appositional growth, chondroblast here on the outer surface, they add new layers to increase thickness. The other thing is that there is the interstitial growth, and in interstitial growth, the cells within this cartilage, they divide, more cells are added. From within these cells divide, and from within, as the cells divide and proliferate, they increase the you can say stroma, or the main material of this cartilage. So this cartilage, it lengthens up, it increases in length. This type of growth is interstitial growth. So two types of growth are there. Interstitial, first we talked about appositional growth, where the perichondrium add more layer, the cellular layer of perichondrium, it adds more layers of the cartilage to increase the thickness, and then second, within this stroma, within this material of the cartilage, the cartilage cells, they divide and they secrete matrix and they proliferate, they secrete matrix. Thus, from within there is increase in the size of cartilage, and hence the cartilage lengthens. There is increase in length of the cartilage. That is called interstitial growth of the cartilage, right? Now, something very unusual and interesting happens. As I've told you, if we focus on here, what is this? This is the perichondrium. So if we focus on perichondrium, as I've told you, it has an outer fibrous layer, which consists of collagen fibers and fibroblasts. Let's not draw the fibroblast. Along with that, we also have the cells called chondrogenic cells or chondroprogenitor cells. What are these called? Chondrogenic cells or these are same thing, basically chondroprogenitor cells. Now these cells, these chondroprogenitor cells, they have unique property. Normally, these chondroprogenitor cells, normally they should transform into chondroblast. Right? But sometimes what actually happens that these chondrogenic cell or chondroprogenitor cell, they transform into a different types of cell, a totally different type of cell, and these cells, these are called osteoprogenitor cell. And what are osteoprogenitor cell? We have discussed them in the last lecture. These are bone forming cells. These are also called osteogenic cell. So this is very interesting and unusual thing. Rather than forming the chondroblast, they transform or they de-differentiate and then differentiate into a different variety of cell, that is osteoprogenitor cells. And of course, osteoprogenitor cell, rather than forming chondroblast, these are osteo, so they will form osteoblast. And osteoblast, what they will do? They will form the bone. So again here, what is happening that this perichondrial cells, the inner layer, the cellular layer of this perichondrium, it transforms into the osteoprogenitor cell layer, and now this perichondrium, it is no more perichondrium, now it is periosteum. And as the periosteum forms, what these cells will do? These osteoprogenitor cells, they will form the osteoblast, and then osteoblast will secrete a bone hair between the cartilage and this periosteum. It was initially perichondrium, now it is periosteum after the transformation of cells. So here a bone collar will be formed. So what will happen that a bone collar will be formed. So here you see bone collar will be formed, and this bone collar, it actually forms with the process of intramembranous ossification that we have discussed in the previous lecture as well. Rather in detail we have discussed in the previous lecture. I will not go into detail here, but the basic idea of course is that the mesenchymal cells in this region, they form the ossification center, and those ossification center will form spicules, and these spicules will coalesce together to form the spongy bone. This is woven spongy bone. Of course, after this spongy bone will be remodeled into this woven spongy bone will be remodeled into the lamellar bone. On the outer side, this lamellar bone will be the compact bone. On the inner side, it will remain spongy, but nonetheless, both of these bones will become lamellar through the process of bone remodeling. Again, that has been discussed in the previous lecture. There is no point of repeating it because this lecture will become very long. It would be already very long, you will see, but with those details I don't want it to become even longer, so people will just skip this video. They will say we will just read the books. We don't want to watch that much huge lectures. Okay, so anyhow, what I was saying that this not peri, this subperiosteal because it is deeper to periosteum. So subperiosteal bone collar has been formed, and as this bone collar has been formed after the transformation of perichondrium cells into the osteogenic cells. Now this perichondrium, it is no more called perichondrium. Now it should be called periosteum. Now it should be called periosteum. Please note that this bone collar, it does not form throughout. It forms in this middle shaft of the bone, which is called diaphysis. Not feces. Feces means something else. Physis. Diaphysis. Let me write it here so you don't get confused. Diaphysis, the long portion of the bone, the shaft of the bone. So here in the shaft region, this bone collar forms. Of course, it's a three-dimensional structure. In this 3D model, it's not 3D model. This is a 2D illustration. So in this 2D illustration, it is looking like that it is two-dimensional, but in reality, if I draw the, let's suppose, section of the bone here, so this is the long bone here. Now this bony collar, it will be surrounding it like this. It will be surrounding it from all sides like this. So I hope you can understand this. It will surround it like this. It's a subperiosteal bone collar. It will surround it like this. So I hope you are clear about this. I don't want anyone to be confused here. If you are still confused, leave the comments below and I will hopefully respond. And by the way, I apologize. Many a times I fail to respond timely or I even fail to respond at all. This is because there are lots of comments and it's hard to respond to everyone. Sorry about that. I try my best to get back to every one of you, but sometimes I fail to do so. So anyhow, let's move forward where we were. Maybe I'm yapping a lot in this lecture. So now let's move forward. So now, as I've told you that these chondrocytes, they are dependent upon the diffusion from the surrounding structures. So the thing is that if they are surrounded, if the shaft is surrounded from all sides with this bony collar, will the diffusion will happen? Can the diffusion happen efficiently? The answer would be no. The diffusion won't be happening efficiently. So that is why these cells may not receive sufficient oxygen. So what will happen when these cells they do not receive sufficient oxygen? Let me draw a cell here, and here I will explain these things to you. So here is the simplified model of a cell with a nucleus. So now what actually happens that there is something called sodium potassium ATPase, not ATPase, ATPase. There is this pump which pumps 3 sodium outside the cell against the concentration gradient and takes 2 potassium ions inside the cell intracellularly, and since this process is happening against the concentration gradient, we need to burn some ATP molecules. And the best way to form the ATP is through oxidative phosphorylation, which happens with the help in the presence of oxygen. Don't ask me what is the oxidative phosphorylation. We will discuss these things in biochemistry. If you don't understand, you can watch some videos on YouTube about the oxidative phosphorylation, or you can refer to your biochemistry textbooks, or you can search on Google as well. So now the thing is that if sufficient oxygen is not present, less ATP will be formed. And as the less ATP is formed, these pumps, they will be underpowered, right? These pumps, they will be underpowered. They won't be efficiently performing their duties to pump 3 sodium ion outside. From inside, there is net loss of 1 positively charged ion outside the cell. So what will happen that as these pumps they fail to work properly, lots of sodium will accumulate in the cell. Lots of sodium will accumulate in the cell. So what will happen? As these salts accumulates in the cell, as these ions rather, as they accumulate in the cell, what will happen that these ions they will pull water with them. And as they pull water with them, the cells they will swell up, they will hypertrophy, they will increase in size. So due to depletion of oxygen, there is hypertrophy of the chondrocytes in this area. So there is hypertrophy of chondrocytes in this area. So here, if I show you the illustration, so you see there is this hypertrophy or increase in size of the chondrocytes in this area. Now these increased chondrocytes, they do something even unusual. Maybe they are upset and they try to commit suicide. Maybe, I'm not sure about this. So what they do is that there is not calcium in this extracellular matrix, in this ground substance. Generally there is not much calcium in this matrix of the hyaline cartilage. So what these hypertrophied cells will do, that they will secrete certain enzymes, for example alkaline phosphatase, certain ions and certain chemicals and certain factors will be released into this area, into this matrix, such that there will be deposition of ions here. There will be deposition of calcium ion, calcium phosphate ions in this area, similar to what happens in the bone. But this process is, remember, this process is not exactly like mineralization of bone. This process is somewhat similar, but there are certain differences that I will not go in this lecture. So basically there is this calcification or mineralization of cartilage here. So as this hard bony type, bone-like, not bony, bone-like cartilage, bone-like matrix forms, then what will happen, that further depletion of oxygen will occur. There is further difficulty in diffusion from the surrounding structure. So there will be further depletion of oxygen, and so much so that these cells may begin to die. But before they die, they do something useful. What they do, that they release certain factors such as VEGF, vascular endothelial growth factor. These factors, they cause the growth of blood vessels in this area. So they themselves, many of them, most of them rather, they die. They undergo apoptosis, programmed cell death, and then there will be, of course, they will leave some spaces here. These spaces are called lacunae. Some authors call it lacunae, these spaces. Some authors call them lacunae, and others, they call it primary areola. Primary areola. From areola reminds me of breast, but these are not breast here. Please don't confuse them with breast. Maybe they look like that nipple area, so that's why maybe scientists put this name. They have certain kind of fascination with breast, maybe. Anyhow, so these are called primary areola, and they are also called lacunae. So now, as I've told you, that these cells, they release certain factors that causes the growth and sprouting of blood vessels in this region. So blood vessels will grow in this region, and these blood vessels, they are now surrounded by a collar of mesenchymal cells and hematopoietic stem cells as well. Let me tell you what are those, and we will discuss the details a little bit later. But the thing is that the blood vessels, sprouting blood vessel, they are surrounded by a collar of mesenchymal cells. Basically, three types of cell may be found in this region. Let me tell you. Mesenchymal cells. These are also called mesenchymal stem cells. And they are coming from this surrounding region, right? Then from the, what is this? The periosteum, which was previously perichondrium. There may be osteoprogenitor cells, And please note that these mesenchymal stem cells, they may directly actually transform into osteoprogenitor cells. That is why in some textbooks they write that mesenchymal stem cells are surrounding these, and some they say that osteoprogenitor cells are surrounding these, and some of the authors they put both. So here I go with the those authors that put the both because it seems more logical, right? And the third thing is that some hematopoietic stem cells are there. Hematopoietic stem cells are those cells from which blood vessels, not blood vessels, blood cells develop, from which blood cells develop. If we go back in the embryology, initially, again, I do not want to delve into detail, but initially you remember that there was the yolk sac, and within the yolk sac, the blood island began to form, and those blood island has these hematopoietic stem cells. But later, the yolk sac regresses a lot. You know, yolk sac, it begins to regress. Yolk sac begins to regress, and some part of it is incorporated into GIT, other part it is regressed. So hematopoietic stem cells, they cannot continue to form in the yolk sac. So later, after few weeks, don't ask me when because I don't remember the dates. You can check it up from the references that you have. So later, from the aorta and aortic region, the region of the aorta, the mesonephric region, so and gonadal region, right? From that region, from the wall of aorta, some hematopoietic stem cells they arise from their aorta, right? But of course, they cannot continue to lie here because aorta is a huge artery. It is the biggest artery of the body and it bears a lot of blood pressure. So these stem cells, they cannot continue to remain here. They need to find some calm place, right? So then later, maybe at ninth week, if I remember correctly, so you can check it up. Later in the liver, these stem cells, they settle, hematopoietic stem cells, they settle in the liver, right? So later they settle in the liver. Right now, you know, the bone and bone marrow, the inner part of the bone, it has not yet been formed, so they remain in the liver. But after the baby is born, right, after the baby is born, initially the function of the detoxification function of the liver, it is taken up by the mother. You know, mother provides the detoxified blood, the blood which is free of most of the toxins, that is provided to the baby by mother with the help of placenta. You know this. But later, as the baby comes out of the body, now the baby has to face the environment, the harsh environment, and from the harsh environment, you know, certain toxic substances are there in the GIT, right? Baby made some certain toxic substances, even the adult as well, of course. So those toxic substances via the hepatic portal vein in adults and even babies as well. Do you remember this thing? So there is this GIT. So from the GIT, this diagram is of course out of proportion. Liver should be here, but this is not anatomically correct diagram. It's a functional diagram. So there is this hepatic portal vein. So everything from the GIT that is absorbed, it first goes to the liver after our master chemist, and our master chemist, it actually detoxify the harmful chemicals here. It detoxify the harmful chemicals that may be present in the GIT, the toxic substances that may be present in the GIT. So now, you know, liver is now the chemistry lab. It is processing the harmful substances. In this harsh environment of liver, in this toxic environment of liver, in this chemical environment of liver, do you think that these hematopoietic stem cells, they can relax and they can easily replicate and they can calmly do their work? I think after the baby is born, now the liver is not the best place for hematopoietic stem cells to remain there. So from here, the liver moved deep into deep into the bone, deep into the bone, into the depths of the bone, into the core of the bone. What is the core of the bone? That is called, here, this is called bone marrow. So after the birth, even before the birth, this process, this transformation starts, that from the liver, ultimately the hematopoietic stem cell, they settle into the core of the bone where they can live peacefully in a special secluded environment of their own, free from toxic radicals, free from toxic chemicals of the liver, so they can calmly remain there and they can calmly continue to produce the blood. So you see the journey. Initially, there was this yolk sac. This yolk sac, as it regress, they come to aorta, right? So these innocent little hematopoietic stem cells, they continue to wander, right? And they first wander to aorta, then here there is huge pressure, so they come to liver. Here there is toxic chemical. Ultimately, they come to their final destination. Here they can calmly do their work, and here they rely. You know, I think we should learn a lesson from these hematopoietic stem cell, that if you want to succeed, you might need to jump from one place to another, from other to the yet another place, and finally, ultimately, if you continue to work hard, ultimately you will settle somewhere. You will settle some calm and peaceful place, and you can then perform your work as intended in that place. Oh my God, I have went too much off topic, but the thing that I want to highlight here briefly is that the hematopoietic stem cells, some of them, they will accompany the collar, and the other hematopoietic stem cell from the liver, they will form monocytes. What they will do, they will form special immune cells, special white blood cells called monocytes, and these monocytes, they will not join the collar. Rather, these monocytes, they will be released in the circulation. They will be released in the blood circulation, and via the blood circulation, they will go inside the blood vessel, and through blood vessel, they will reach here. And by the way, these mesenchymal collar, these cells, they will also come within this and spread within this region. And the monocytes, they will also, you see, these are monocytes, they are coming from the blood, they are inside the lumen of the blood vessel. Please remember that some of the cells, mainly mesenchymal cells and osteoprogenitor cells, they are outside, they are in the collar, right? But monocytes, they are inside, they are within the lumen of this blood vessel, right? So here they will reach here, and here what they will do is that multiple of these multiple monocytes, they will unite together, they will fuse together to form a single huge monstrous cell, giant cell. And this giant cell has multiple nuclei. This is called multinucleated giant cell. Basically, it's a type of a macrophage. It's a type of macrophage. This specialized macrophage, what it will do, that it will come here and it will digest this calcified matrix, which is originally created by the chondrocytes. Generally the calcified matrix is created by which cells? It is created not by chondrocytes or chondroblast. It is actually created by osteoblast or osteocytes. It is actually created by osteoblast. But here it was created by those dying chondrocytes, right? So, you know that those macrophage cousins, macrophage-like cells that dissolve the calcified matrix of bone, these are called osteoclasts. But here, because they will be dissolving the cartilage, these are not called osteoclasts, these are called chondroclasts. But the thing is that these cells, these are exactly like osteoclasts. There is no difference histologically and histochemically. There is no difference between chondroclast and osteoclast. So some authors, they write like this. They write it like this. They write that there is the entry of chondroclast or osteoclast because they are same cells. They are functionally very similar. They are structurally exactly same. They are identical, right? But the only difference is that osteoclast, they dissolve the calcified matrix created by the osteoblast, but chondroclast, they delete they remove the calcified matrix created by chondrocytes. There is no other difference, right? So now what will happen? Here I will not go into detail. We have already discussed these things previously. Now, of course, I have shown only one cell, one such cell here. There will be multiple such cells. Now, these cells, they will, at some places, they will remove this matrix. They will remove this matrix, and then these mesenchymal cells or osteoprogenitor cell, if there are mesenchymal cell, they will transform into osteoprogenitor cell. If there are osteoprogenitor cell, they will transform into chondroblast, not chondroblast, sorry, osteoblast. So gradually, chondroclast, they will remove the calcified matrix, and osteoblast, they will form the bone. Now please note that this is not a sequential process. It's not like that this matrix will first be dissolved by chondroclast and then whole of the bone will be formed in this region, but it is sort of asynchronous process, that first in this region, let's say this hair is removed, at other point here the matrix is removed, at other point matrix is being removed, at the same time the bone is being formed. So you see, at multiple places there are the formation of these small little ossification centers, and these ossification center bony spicules will be formed similar to how we saw in the intramembranous ossification. So multiple bony spicules, some will form here, some will form here, some will form here. They will coalesce to form a proper network of woven spongy bone, as we had discussed in the last lecture, right? So again, ultimately the spongy bone will form here like this. So the spongy bone will form here, right? So woven spongy bone will form here, right? And in the core region, what happens that either the bone never forms, I'm not sure which one is correct. Either the bone never forms, or if it forms, it is dissolved by the osteoclast. It is dissolved by the osteoclast in this region. And here, of course, the mesenchymal cells and hematopoietic stem cells will come here, right? And these cells, what they will do, that they will form the bone marrow here. They will form the bone marrow here. And one thing that I forgot to mention previously, that is very important, and I should probably go back and tell you the name of that. Now here you see that this blood vessel, it is surrounded by the collar of mesenchymal cells. It is surrounded by this collar of mesenchymal cell. Now whole this complex, the blood vessel along with the mesenchymal collar, whole this complex, what it is called? It is bud-like structure. It is called periosteal bud. It is called periosteal bud. Of course, I haven't shown here, there will be a bigger artery here, and from the artery, of course, there is the formation of this bud. This bud actually arises from a larger artery here, and of course here this bud will be surrounded by the mesenchymal collar. This I forgot to mention. This is very important point, by the way. So yeah, that was about it mainly. So you see that the bone, it forms in the diaphysis. I don't know why I keep on calling it diaphysis. It's diaphysis. So this spongy woven bone, it forms in the diaphysis region. Of course, it will be transformed, it will be remodeled into the lamellar bone, as I have discussed in the previous lecture. It will be remodeled into lamellar bone, and here this bone, it will be remodeled into compact lamellar bone. This will remain spongy, but of course it will become lamellar, but here it will transform into compact lamellar bone. Now what about the other part of the cartilage? It is not yet replaced by the bone. You see now you got the concept, hopefully, that this cartilage, its cells were dead, its matrix was dissolved, and it has not been replaced by the bone, and that bone actually formed with the intramembranous ossification. So cartilage is basically act as a scaffold, acting as a scaffold, or it is acting as a mold, or it is acting as a cast, or it is acting as a template, whatever you like to call it. But in the end, actually, the intramembranous ossification is going on here. So what we can say that truly speaking, technically, the endochondral ossification is a more advanced and more modified kind of intramembranous ossification, more sophisticated type of intramembranous ossification. It is nothing more than that. Here the cartilage is acting as a scaffold. For the simpler bone, for the simple flat bone, we don't need such type of cartilage or scaffold. We can simply form them directly from the mesenchyme, directly from the mesenchymal condensate. Here we need a scaffold, here we need a cast, and for that this cartilage has to be first formed and then later it is replaced by the bone. Now what about these ends? The ends of the bone, these are also called epiphyses. So here the story will repeat that again periosteal bud will form. I will not show you the story again that periosteal bud will form and they will again form this bone here. What is this? The spongy woven bone, which will be again replaced by the lamellar bone gradually by the process of bony remodeling. Now, let me tell you three important points here. This is almost the end of the discussion, but here let me tell you some important points. Number one, you can see that some of the cartilage is never replaced in here. And what is this cartilage? Let me tell you. So here, the surface of the bone, it is still covered by, this is basically articular surface. Here there will be another bone, so this is the joint articular surface here. So this articular surface, it is covered by a cartilage which decreases the friction here, right? So it sort of protects the bone and it decreases the friction here. This is called articular cartilage. And it is the remnant of original fetal cartilage that formed, original fetal hyaline cartilage. And of course, there is no fun in telling that this is the hyaline cartilage. It's very logical. Here as well, here also, here there is again the articular cartilage. The second cartilage that you see here as well as here, and they may be more than two as well. So these cartilages, what are they called? These are called epiphyseal cartilages, and these are also called growth plates. What is the significance of them? Again, it's a whole lot different topic that I won't go into in this lecture, but the benefit of this epiphyseal cartilage is that this epiphyseal cartilage, it will proliferate, it will increase in size, and it will gradually be replaced by the bone, right? It will be gradually be replaced by the bone, and in this way throughout the life, not throughout the life, till adolescent, right? Till the person reaches the adolescent, it will help in increasing the length of the bone, and ultimately as this kid, as it reaches the adolescence, this epiphyseal cartilage which will also ossify. It will also ossify ultimately, and this will ultimately be replaced by the bone. How this process occurs, that is beyond the scope of this lecture. But it is worth mentioning here that epiphyseal cartilage and articular cartilage, these are basically the remnant of original cartilage. Okay, this was the point number one about remnants of cartilage. Now the second point that I would like to mention here is that what if I told you that this discussion of that I've told you about the oxygen depletion and hypertrophy of cell, this discussion might be totally wrong. But doctor has asked, if it is wrong, why did you waste our time with so much details of oxygen depletion and ATP and stuff like that? Well, first let me tell you why I think it is wrong, and I'm not alone to think that this is wrong. Actually, you know that the subperiosteal bone collar forms in this diaphysis region. diaphysis region. But no such periosteal or perichondrial bone collar. It does not form here in the epiphysis. It does not form in the epiphysis, but still this epiphyseal region, it also undergo the similar phases as the diaphysis. Here also the cells, they undergo the not atrophy, hypertrophy, they swell up. Here also the cells swell up. Here they also secrete the calcified matrix. Here also the periosteal bud forms, and here also the bone forms very similarly as in diaphysis. Here also the bone forms very similarly. So it means that there is a possibility that either this oxygen depletion theory is wrong, or it may not be very significant. It may not be very significant. There may be some role of it, but it may not very significant, right? And rather than this oxygen depletion, the genetics may be important here. Not genetics, I should say the transcription factors would be important here. Certain transcription factors are released in a proper sequence, which led to hypertrophy of these cells, followed by death of these cells, I mean chondrocytes, followed by first there is hypertrophy, then there is calcification, then there is death, then there is the periosteal bud and stuff like that. I hope you got the point. But still, many authoritative textbooks, they mention this oxygen depletion theory. For example, one of the very famous book of what is this? Histology, Janquera histology, it mentions this theory. So, or hypothesis, whatever you like to call it. So this oxygen depletion hypothesis, it may not be very significant, or it may be even wrong, but at least it helps you remember the sequence of events that is going on here. So 2 point we have discussed. The third point I have already discussed, that endochondral ossification is basically a modified or advanced type of intramembranous ossification, in which the cartilage is acting as a mold or template or cast or scaffold, right? So that was about the endochondral ossification. Finally, let's have a quick review. So first of all, there is mesenchyme, then there is the formation of mesenchymal condensation, which roughly resembles the shape of the bone. Then we have the formation of hyaline cartilage, and then this hyaline cartilage, it grows in size via the appositional growth as well as interstitial growth, and of course the hyaline cartilage, it is covered by the perichondrium except at the articular surfaces. Then there is the formation of there is conversion of the perichondrial cells, the chondrogenic cells in the perichondrium, they are converted to osteogenic cells, which forms the subperiosteal bone collar via the intramembranous ossification. That leads to hypertrophy of the cells, hypertrophy of the chondrocytes, and these chondrocytes, they begin to die, leaving the primary areolas. And by the way, as the osteoclast, they start to eat up the what is this, calcified matrix of the cartilage. Now the primary areolas, they are converted into secondary areola. So these were the secondary areola. So now the periosteal bud forms, which carries the mesenchymal cells as a collar, and inside the lumen there are monocytes which unite, which fuse to form the chondroclast or osteoclast. So chondroclast or osteoclast, they begin to eat up calcified matrix to form even larger spaces, even larger lacunae. These are called what they are called? They are called secondary areola. So secondary areola are formed, and these secondary areola are filled by the bone formed by the osteoblast, osteoprogenital cells, mesenchymal cells converted into osteoprogenital cells, which in turn are converted into osteoblast, which form the bone, right? So again, here the bone is also formed by the process of intramembranous ossification. So bone is formed here, then in the core, this bone is dissolved, and the hematopoietic stem cells that came here and mesenchymal cells that came here, they form the bone matrix. They form the bone, not bone matrix, they form the bone marrow. I'm sorry about that. This is not bone matrix, this is bone marrow. Please correct it. So they form the bone marrow. Then secondary ossification centers they form. You see in the epiphyses, epiphyses are like that before the birth, right? These epiphyses, they begin to ossify after the birth. Please note it down. They begin to ossify usually for most of the long bones. They begin to ossify after the birth. So this was the primary ossification center. These are the secondary ossification centers. And by the way, still the articular cartilage and hyaline, not hyaline cartilage, of course it is hyaline cartilage. There is epiphyseal cartilage or growth plate. They remain cartilaginous until the adolescence. So here is again the complete review: mesenchyme to condensate, mesenchymal condensate to cartilage to then growth of cartilage, then formation of subperiosteal collar, then formation of then hypertrophy of chondrocytes, then death of chondrocytes, then periosteal bud formation, then formation of bone in the diaphysis, then formation of bone matrix, finally after the birth epiphysis. That is also ossified. And again, all these bones that form here, that they are formed with the help of intramembranous ossification. And please don't forget that ultimately this bone, it will be remodeled. As we have discussed in the last lecture. Very similar to that, remodeling of bone also occurs here. So I think that was all about the endochondral ossification of the bone. Thank you so much for watching this video. Have a great day. Goodbye.